Hi everyone. In this lecture, we're going to continue discussing about gene action. So we've talked about what genes are and what genes do, and now we're going to talk about what happens when there are mistakes. We're going to talk about a topic called gene mutations. So in this lecture, we're going to be discussing the different types of mutations, and we'll also discuss how those mutations can occur. All right, so we talked about a gene, and we know that a gene is a specific sequence of nucleotides, right? And those nucleotides are then used to create those. We have those codons, right? We copy that into messenger RNA. We read the messenger RNA one codon at a time. It brings in a series of amino acids, which then we fold and make a protein. Well, sometimes in that process, and it's quite a complicated process, mistakes happen. And when mistakes happen, we refer to it as a mutation. So mutations are changes in the nucleotide base sequence. So in the DNA itself, we have errors that occur, and a C gets turned into a T, or a couple of letters go missing, or whatever happens to happen. We'll talk about all of the types of mutations in just a moment. But it's important to note that mutations aren't necessarily bad things. Yes, there can be some harmful consequences of mutation, but they're also an essential ingredient for life. So mutations are the ultimate source of variety in any population. So if you think of human populations for a moment, and you think of the fact that at one point in human history, all humans had darker skin, dark hair, and dark eyes. Through a series of mutations and moving patterns of human beings over the course of history, we saw those physical characteristics change due to mutations. We saw light-eyed mutations, such as blue eyes and green eyes. We had lighter skin mutations. We also had some darker skin mutations. So all of those things had an advantage, and they gave those individuals who had those mutations an advantage as far as being able to survive and reproduce. And that's getting a little into evolution right now. But what's important here is that, again, mutations are the source of all of the new varieties of phenotypes that we see in a population. So mutations are essential for living things to be able to survive changing environments. So how do we define a mutation? It's technically a change in a sequence of DNA that is present in less than 1% of a population. So mutations are rare to a certain extent. That's how they start off with. However, and this can occur again at the DNA or the chromosome level. However, if that mutation becomes more common, if that genetic change is found in greater than 1% of the population, we don't call it a mutation anymore, we call it a polymorphism. So it takes on a new identity. It's not a rare thing. It begins to confer some advantage to the individuals who have it, and so therefore we then begin to refer to it as a polymorphism. It's a new gene variant, if you want to think of it that way. And as I mentioned before, effects of mutations can vary. We sort of have this negative connotation with mutations that they're all bad. And there are certainly bad mutations and mutations that cause genetic diseases. But there are also mutations that can essentially confer advantage or give individuals an advantage. So we have something called loss of function mutations or gain of function mutations. Typically, again, these are things where we have a protein that doesn't work anymore, or a protein that is working, we're producing too much of it. That could be another example, if you will. And again, depending on what the overall outcome of that mutation is it harmful? Is it beneficial? Is it neutral? Kind of depends on how we sort of frame our discussion around that particular type of mutation. So if you've heard the word mutant, it just refers to an individual who has that phenotype that is, again, the mutated phenotype. So again, something that's rare. And again, mutant in sort of conventional terms usually denotes something that is weird or bizarre or abnormal. But it's uncommon, but it doesn't necessarily mean that they're abnormal in some way. It just means that they have this, again, this mutation. All right, so let's go into some different types of mutations for a few moments. So the first one I'm going to talk about is the simplest one that we can think about, and it's referred to as a point mutation. And in a point mutation, we substitute one DNA base for another. So here's our original codon. So here's our codon right here, C8T. We're just going to sort of look at this part here. Okay, the second part of the strands, remember we have the coding strand and the non-coding strand when we were talking about protein synthesis. And then remember in DNA replication, we use both sides as templates. So let's say this is our template and we are going to make a copy of it. Well, as we're making a copy of it, we forget to proofread this mistake. And instead of this A pairing with a T, which is what it's supposed to do in our replicated DNA, a C accidentally gets substituted in there. Okay, so now we've got a point mutation, a single error. So here's the possible outcomes. Okay, we could have our original DNA molecule, okay, the one side that got copied, which has the original information, but the other side of our molecule is now going to get copied over and over and over again, and it's going to be wrong, right? We're going to have this sequence now that's a C and a G once it's copied, and so here we've substituted what was a T pair, and now we have a GC pair. So that's our point mutation. Very simple. There's two types, what we call transition point mutations and transversion point mutations. So a transition point mutation was when a purine replaces a purine or a pyrimidine replaces a pyrimidine. So if you remember, adenine and guanine are purines, and thymine and cytosine are pyrimidines. Okay, so if you substitute one for the other, it's transition. If you substitute the A with the T or the C with the G, it becomes what's called a transversion. Next one I'm going to talk about is a missense mutation. So missense mutation, so we're still, I guess, let me back up. We're still talking about point mutations. What happens when you have a point mutation? A couple things could happen. We could have what's referred to as a missense mutation. This is a type of point mutation where we're changing one amino acid into another amino acid. Okay, so here was our original sequence, right? So when we go through the process of transcription and translation, we would end up with these amino acids, okay, methionine, glutamine, alanine, and glycine. If we change this original DNA triplet here, GAA, and actually change the A to a C, GAC, now we've changed the sequence of amino acids. And now it's methionine, aspartame, alanine, and glycine. Okay, so we've changed it, and it's called a missense. There's also something called nonsense mutations. And nonsense mutations are when we change an amino acid into a stop codon. So in a stop codon, we would absolutely just stop making that amino acid. So the missense mutation is the one that is sort of the smaller mistake, right? You're substituting one amino acid for another. It might have a relatively minor impact on the protein that we're making. It might have a major impact on the protein. It kind of depends on the protein itself. The nonsense mutation is much more harmful because you're stopping production of the protein in and of itself. You're cutting it off before it's finished. And so this is usually a more dramatic consequence of a mutation. All right, let's talk about the next type. So those were point mutations. Let's talk about frame shift mutations. Frame shift mutations are different. In this case, we are shifting our reading frame by either adding a base where it doesn't belong or deleting a base where we shouldn't have done. So let's go back to our original example from before where we had methionine, glutamine, alanine, and glycine. In this case, we're going to remove this G right here. Okay, so we're going to take this out. It didn't get copied. A mistake happened. Now what you see is only methionine stays, and now we have lysine and histine instead. So every other amino acid throughout the entire rest of this protein is going to be wrong. So again, a major consequence, even though we're just adding or taking away one base, it has a huge consequence on the production of that amino acid. The only way you can sort of mitigate that is if you have a deletion that actually removes 3 bases or multiples of 3. In that case, you wouldn't be shifting the read frame anymore. You're not changing all of the amino acids. You're just going to be missing a single amino acid or 2 or 3, depending on how many bases got deleted. And then conversely, if we duplicate a part of a gene, if we copy it an additional time, we would have more amino acids in our sequence than we need, and we would have a part of that amino acid sequence that actually is replicated. All right, so I sort of have this mutation analogy here where we're going to look at these different mutations we talked about: missense, nonsense, frame shift, deletion, insertion, and duplications, and we're going to relate it to a sentence here. Okay, so our normal DNA sequence would be like a sentence that reads, "The one big fly had one red eye." And what we're looking for is how different the sentence is after each of these types of mutations. So a missense point mutation, where we're just changing one amino acid. In this case, we're going to change the letter Q, but you can still kind of read the entire sentence, even though it's THQ, one big eye, fly had one red eye. Okay, you can still kind of read it and understand it. In a nonsense point mutation, we're going to stop the sentence altogether. The one big, we don't know what the rest of that is, so we can't finish the sentence. In the frame shift mutation, again, you have a big change. The one, let's insert a Q where it doesn't belong, and now none of the sentence makes sense. Okay, deletion has a missing word. Insertion has a word that wasn't in the original sentence, and duplication has a word twice. So again, making the connection now between what we're looking at and the proteins themselves, missense point mutation, that protein's probably fine. Nonsense point mutation, nope, doesn't do its job. Frame shift mutation, nope, that protein probably can't do its job. Deletion, insertion, and duplication mutations, it sort of depends on the size of the deletion, insertion, or duplication. It's possible the protein function won't be affected negatively, but it's also possible that there will be small consequences of those mutations. All right, which of the following statements is false? Let's do a quick comprehension check. Mutations are always harmful. A mutation is a change in the DNA sequence. A mutation is present in less than 1% of the population. A mutation may result in the overproduction of a protein. If you remember, it's number one. Mutations are always harmful. They're not always harmful. There can be other consequences of mutations as well, which we will discuss. All right, so mutations can occur in two different types of cells. If you remember, we talked about germ cells and somatic cells. Germ cells were the cells that we were discussing in our conversation about meiosis, and those are our reproductive cells, right? The cells that make the egg and sperm in males and females. So in a germline mutation, if a mutation in the DNA sequence occurs during DNA replication before meiosis begins, that gamete will be affected and all of the other cells will also be affected. So what that means is that a mutation in a germline cell will affect that potential offspring. So if that gamete, that egg, or that sperm goes on to fertilization and becomes a zygote one day, every single one of their cells will have that mutation. That is as opposed to what's called a somatic mutation. Somatic cell mutations only occur in certain cells in your body, and the only cells that are affected are the ones that happen in mitosis. So if it happened in a skin cell, only the new skin cells produced from that skin cell would then have that mutation, or the liver, the heart, or wherever it happens to be. Okay? This is also how cancers happen. If you guys remember when we talked about there are errors in the checkpoints, those errors in the checkpoints are caused by mutations in the genes, those checkpoint genes. And if you have enough of those errors adding up to cancer and the cell begins to divide uncontrollably, every cell that it makes a copy of will also have that mutation. So how do mutations occur? They can be completely spontaneous, or they can be caused by exposure to substances, chemicals, radiations, etc. Anything that's capable of causing a mutation, a substance that can cause mutations, is referred to as a mutagen. So a good example of mutagens would be radiation. We know all types of radiation are mutagens. Okay, they can cause mutations in our DNA. So it doesn't matter whether you're talking about an MRI or an X-ray or radiation coming from the sun. All forms of radiation have the ability to make our DNA fragile, basically, and cause mistakes. So that's why it's always recommended that you limit the amount of radiation you're exposed to, whether you're talking about medical radiation or environmental radiation. So what happens in these cases, we have something called spontaneous mutation. So again, we can have exposure to things that that make our DNA do this, but this can just happen spontaneous and randomly. And we're all walking bags of mutations. So spontaneous mutations occur about 1 every 100,000 bases that are copied. A mutation is made. So you copy 100,000 of your DNA bases, and you're going to make a mistake, which is a pretty good ratio if you think about it. That's not bad. Most of those mistakes are caught, right? So we catch them, your body catches them, and it repairs them, and it's not a big deal. But some of those mutations aren't caught. So here's what happens is we're going through the process of DNA replication, and we have that DNA polymerase that's reading the sequence of bases, and it makes a mistake, and it brings in the wrong pair where it's not supposed to. That's how mutations occur. So again, spontaneous mutation rate, it differs between genes. Large genes have higher mutation rates, but again, each human gene has about a 1 in 100,000 chance of mutating. So again, you copy 100,000 base pairs and you're bound to make a mistake. And each individual has multiple new mutations. I happen to have a mutation that makes me immune to chickenpox. I've always been immune to chickenpox. I never had the chickenpox vaccine. It didn't exist when I was a kid. I've always been immune to chickenpox. I know my mother is also immune to chickenpox. She never had it either. We don't know how far back that mutation goes. She can't remember if her mother was immune to chickenpox or not. But in any case, that's a mutation that I'm walking around with that's relative. So there are also certain places in our genome that we refer to as mutational hotspots. And in mutational hotspots, these are places where we're more likely to make mistakes. And we typically tend to make mistakes in any sequence that are short and repetitive. So imagine you're typing a paper, okay, and you're transcribing. So you're looking at a piece of paper and you're reading the words and then you're typing what you're seeing. So imagine if you were copying these short little phrases on the paper over and over and over again, right? So he went right, he went right, he went left, he went left. And let's say that just goes back and forth, over and over and over again. You're a lot more likely to make a mistake there because of the repetitive nature of that sequence. The exact same thing happens with your DNA. If you have a short sequence, say TTAACGT, and you repeat that 50 times, it's probably likely that your DNA is going to make a mistake. Again, and so we sometimes miss those because the repair enzymes don't catch them. The other place we see hotspots are in places where we have palindromes. So remember, palindromes are sequences that can be read forwards and backwards, and they're identical regardless of which way you read it. So these are often associated with insertion or deletion mutations. All right, so other ways that you can be exposed to mutagens. So again, we have these spontaneous mutations occur, and then there are actual instances when you're exposed to mutagens that make your DNA susceptible to those mutations. One is induced. So this would only be for scientific study. It's not really done in humans. We do this with fruit flies and mice and things. We want to see what the effects are of certain chemicals and substances on cells and disease and things like that. So we could do it intentionally for a purpose. Accidental exposure is someplace you could be exposed. So in a workplace, in industrial accidents, again, medical treatments. So your MRIs and your CAT scans and your X-rays, all of those types of things. Any type of exposure to radiation, essentially. And then, as I mentioned, natural exposure. So sunlight, cosmic rays, minerals in the earth. There's something called radon that's a gas that basically comes from the ground. It seeps from the ground naturally, and radon is a mutagen. It's a known mutagen. It can cause lung cancer. So again, a whole host of places you potentially could be exposed to radiation. Let's say you have a thermonuclear power plant. If there was a core meltdown and some of the radiation escaped, you could possibly be exposed there, like the people of Chernobyl were back in the 80s. So, the effects of mutations. 3 things can happen. They can be harmful, they can be beneficial, or they can be neutral and nothing can happen. So, harmful mutations. This is the way that disorders and diseases, genetic disorders and diseases, this is how they happen. It's when proteins can't do their job because they aren't formed properly. There can also be beneficial mutations. So again, we don't notice these very often, but these are often the ingredients to leading to evolving populations. So they are better and stronger than those individuals. I shouldn't say stronger. They are better and more fit than those individuals who don't have those mutations. And I'll talk about a couple of those in a minute. And then largely by and by, you just have neutral mutations. Nothing happens. So this is what happens the vast majority of the time. Nothing at all happens. Remember the redundancy we talked about in the code, how we can make changes and there were multiple codons that would code for the same amino acid? That's great, because if we do make mistakes sometimes, it doesn't matter because it doesn't change anything. The protein still works just the way it was meant to. Okay, quickly sort of wrapping things up, some beneficial mutations that you should probably know. So if you can drink milk, thank a mutation. Beyond infancy, no mammal is supposed to drink milk. Milk is something that only infants are supposed to drink from their mothers, and then they lose the ability to digest milk because their bodies stop producing lactase, which is the enzyme that breaks down milk sugars. 10,000 years ago or so, in a couple of different populations, we had an independent mutation occur, which enabled our bodies to continue to make lactase. And because we can continue to make lactase, we could then begin to drink milk and eat milk products. So it was a huge boon for human populations because we had a new source of protein that we didn't have before. We didn't always have to kill our goats or our sheep or our cows for protein. Now we could use their milk to make things. There are individuals who are naturally resistant to HIV. So their cells are not susceptible to the virus that causes AIDS, the HIV virus. It can't penetrate their cells, and so therefore they can't be infected. Something called resistance to atherosclerosis. Atherosclerosis is sort of a narrowing of the artery because you have all these plaques that form in it. Individuals have a mutation that the plaques don't form the same way. And then depending on how you feel about twins, hyperovulation is actually a mutation. So women sometimes will release more than one egg per menstrual cycle or per cycle, and when they do that, twins can occur. All right, final comprehension check. A mutation may occur due to which of the following? Exposure to X-rays, exposure to cigarette smoke, environmental disasters, or all of the above. All of these things can lead to mutations. Okay, here's wrapping things up, important terms you should know and concepts.